In the fight against specific blood cancers, immunotherapies like Yescarta and Kymriah are delivering results once thought impossible, remissions that last a decade or more. CAR-T is a living drug. While a traditional chemical pill is inert and shelf-stable, these engineered cells are reactive and highly sensitive to their environment. Producing custom human biology for thousands of people is a logistical challenge that requires two independent manufacturing pipelines to run in parallel. Track 1 is the production of a viral vector, the delivery vehicle engineered to carry the specific genetic instructions used to hunt cancer. Track 2 involves the preparation of the material for those instructions, the patient's own immune cells. To understand how this treatment actually works, we have to map out the scale of these two production tracks before they finally collide. To get genetic instructions into a cell, manufacturers use lentiviral vectors derived from the HIV-1 virus. Safety is the primary requirement. Engineers strip out dangerous regulatory genes to create a self-inactivating vector, ensuring it delivers its payload without replicating. Moving from a lab to global distribution requires industrial volumes of this viral vector. The traditional method relied on growing host cells in flat, single-layer plastic flasks, where the cells needed a physical surface to attach to and grow. To meet high demand, the industry adapted these cells into suspension cultures. These cells no longer need an anchor. They float freely in liquid. This shift allows manufacturers to grow high concentrations of the virus in controlled, single-use bioreactors that hold up to 2,000 liters. These suspension bioreactors transform viral production from a slow, manual process into a standardized industrial operation. The liquid harvested from these bioreactors is a raw soup, filled with the virus, but also dead host cells and cellular debris that must be removed. The first step is centrifugation. High-speed spinning forces heavier cellular debris to sediment to the bottom, leaving the lighter virus in the liquid above. The remaining fluid passes through microfiltration membranes to catch any microscopic fragments the centrifuge missed. With debris cleared, manufacturers use nuclease enzymes to digest residual DNA left over from host cells. This level of purification is mandatory. Even trace impurities could trigger severe inflammatory reactions once the treatment enters a patient. While the viral vector is processed in a factory, Track 2 begins in a hospital, where the patient is connected to an apheresis machine. The machine draws the patient's whole blood and uses centrifugation to separate it into distinct layers based on density. The target is the buffy coat layer, which contains the lymphocytes needed for the therapy. Unlike factory-standard cell lines, a patient's blood is highly variable. Many have already undergone chemotherapy, leaving their remaining immune cells weakened. To protect this fragile material during shipment, clinical staff cryopreserves the cells, freezing them at temperatures low enough to halt biological activity. Once the material reaches the lab, it is thawed and put through separation processes like illustration to isolate the specific CD3-positive T cells required for engineering. Track 1 is a feat of industrial scaling. Track 2 is the opposite, the careful preservation and isolation of a single person's biology. This is the moment where the tracks meet. The mass-produced viral vector is introduced to the patient's isolated immune cells. Through transduction, the vector integrates its payload into the cell, permanently writing a new genetic sequence into the T cell's genome. This code directs the cell to grow a chimeric antigen receptor, or CAR. The blue binding domain on the exterior locks on to the orange cancer antigen. A signal then travels to the purple signaling domain inside the cell membrane, triggering its lethal immune response. These engineered cells are expanded until they number in the millions. They are now uniquely programmed to hunt down the patient's specific cancer. CAR-T is a triumph of supply chain engineering. It proves that we can rewrite human biology for an individual and scale that process to meet a global need.